I'll talk about convergence and reproducibility and results we've gotten on the Blue Waters resource. Various people in our lab, and we use computer time not only at Blue Waters, but all the way back to resources locally at our university, so a lot of data transfer. When we start talking Blue Waters and allocations, they're in the order of 7 to 14 million node hours. It's actually really difficult to use these resources to get students that are willing to put in the time to get these large-scale applications going, and we'll talk about some of these challenges. And what I want to do is model the structure of RNA as accurately as possible. It requires good balanced force fields, fast, accurate methods, good experiments to assess the results, because you can simulate anything, and unless you can compare it to some form of reality, like an experiment, you can't tell if it's real. We want to be able to get full dynamics and complete sampling of the thermally accessible conformational ensemble. And to reliably do that, we got to understand the convergence. Are we getting the complete sampling of the conformational ensemble, and are the results reproducible if we start from different initial conditions? And RNA is kind of tricky because various things can interact with it, like ions or drugs or other biomolecules, and alter the structure. There's also an inherent dynamics. So you may select particular conformations that are dynamically sampled, or you may induce a particular conformation, and it's hard to assess dynamic structures experimentally. And what we often saw in the simulations over the last 20 years, that is, if you run a nice short simulation, you stay very close to the experimental structure, and as you more fully and completely sample, you move away from experiment to something that's more favored and hence wrong, so the methods are inaccurate. But I'm starting to see now light at the end of the tunnel. We're starting to see some success in the results. This is from a slot canyon in southern Utah. Normally when I give my talks, it's kind of gloom and doom about how well we do, but now I'm starting to get pretty excited, as we'll hear today a bit. So one interesting simulation case is there's a loop structure of RNA that is different when ions are present, like magnesium, or when they're not present. And we can throw a magnesium molecule into these simulations. This is the piece of the RNA. You don't see the water and the ions that are there. But if we add in magnesium, we can convert to that correct structure, which is pretty exciting. And the main tool we use is Amber. It's been around for a long time. I don't want to spend a lot of detail discussing it. We just released a new version of the open source Amber tools, which contains all the Amber codes except for PMEMD, which is a fast molecular dynamics engine on GPUs and on CPUs. But like all the QM code now is open source, and that's just come out now. One of our features is an extreme performance on GPUs, which I'll talk about a bit more. So I already mentioned this. If we run a short simulation, you tend to stay near the experimental structure. Invariably, when we went further, we went to something new. Your hope is that this new structure is higher energy than what we know from experiment, and you'll come back eventually, but often the case, it turns out this anomalous structure is in fact more stable than the native. And we could sample these in various different ways. We could use, like David Shaw's Anton machine, which can do 16 microseconds worth of movement or dynamics of one of these molecules in a day, super fast. And if we look at this RNA tetraloop, which is super stable, I ran these simulations probably about a decade ago now for a microsecond and a half, and it looked like it was perfectly stable. That took like 6 months. When we went to Anton, I got the same microsecond in 100 minutes on that resource, but it blew up in the simulation because the force field had issues. Now, we don't all have access to Anton, and no one can get access for more than 1 week unless you start to work for David Shaw. So we want another way to do it. So another way to do it is to try to look at ensembles of independent simulations. And ideally, if you can aggregate the results together, and the length of these shorter simulations captures the process of interest, you should be able to get the same kind of results. And the nice thing about our performance on GPUs is even on Blue Waters and the K20 NVIDIAs, we get about 200 nanoseconds a day. So compare that to 16 microseconds, we'd need about 80 GPUs to match that if we can do that, and that's a hell of a lot cheaper than buying an Anton machine. This you can't see from the anywhere in the room probably, but the black is performance on a Sandy bridge for our current code. It's about 30 nanoseconds a day. K20, 200 nanoseconds a day. If we go up to the K80s or K40s, you can go even faster. The best performance is on 4 K40s right now. And there's work underway to get us to scale to 8 GPUs. as well. Now, if we run these independent simulations as an ensemble instead of a single instance on Anton for this RNA tetraloop, a number of the simulations look fine over this 200 nanosecond timescale, but a number of them are blowing up. Essentially, they're moving away from that correct experimental geometry. And I would contend if we ran this even longer, they all would effectively go away and never come back. Same thing on Anton. And so there's issues we got to look at in terms of how converged are the results and how reproducible, and what we can do to sample. I've talked about the DNA duplex convergence before, and we've published papers on it, so I'm only going to blast through a quick point here. But we could run long simulations on Anton. We did this back in about 2001 or so. Smooth trajectory of the DNA, you don't see the water and the ions. And what the results showed us, which was really counterintuitive to me at the time, was if we take a time average of the motion of the DNA over, say, a 3 microsecond time scale from different windows of the trajectory and superimpose the structures, they were essentially identical, that they were no longer moving. So what that means is, on average, if you look at a 3 microsecond time scale, the DNA is effectively static. On a picosecond time scale, the fluctuations are huge, and the fluctuations in groove width are bigger than the sequence-specific differences in groove width that you would see in this static microsecond time scale structure. So we reran all these simulations thinking this was a bug on the Anton machine. We ran them on Blue Waters and on Keeneland as ensembles and got the same results, and then started to think about it and realized that experimentally, there's very few experiments that show motion in Watson-Crick DNA on the 1 to 5 microsecond time scale. Those motions are absent. It was claimed that those can't be seen because they're hard to measure experimentally. But it turns out if you pop a mismatch in here, or even a Hoogsteen base pairing, suddenly a signal appears in that microsecond time range, and that's probably important for DNA damage recognition. And also the time scale of encounter of molecules with DNA is on the order of a microsecond or so, so the DNA is showing effectively a static structure on that time scale. Okay. So one thing we have to do is show the reproducibility of the results. So if we run simulations on Anton or on a CPU or on GPUs, do we get the same results? And one way to do that is to combine the results. We have these structures as a function of time from two different simulations. You combine them, you calculate the modes of motion for that, and then you separately project those modes of motion on the independent trajectories. If the trajectories sample the same motions, there should be an overlap in those modes. And this is shown in this plot where there's a dotted line and a solid line that are exactly on top of each other. They sampled exactly the same modes of motion in the two independent trajectories. So we've got this reproducibility and effectively convergence in the internal portion of the helix. We don't have base pair opening converged yet. I'll skip that for now. So, you know, we were running a lot of simulations on lots of different RNAs and DNAs, and they all kept blowing up. And we made improvements to the force field, and we could run a little bit longer. We could suddenly run, you know, we get access to Kraken. We could run for 500 nanoseconds. We'd run for 600, it'd blow up. We go fix the force field, and then we get the GPUs. We can go to a microsecond, it blows up, keeps blowing up. So maybe these systems are too hard to look at. So we want to look at something simpler. We moved to just this simple tetranucleotide. It's only 4 nucleotides. It's got a precise experimental measures of 2 structures that are sampled experimentally, what they call the major conformation and the minor, about a 3-to-1 occupancy. And we can now completely sample this thanks to Blue Waters. If we just do a straight regular molecular dynamics simulation, colors are different clusters we sample. Even at 5 microseconds, we haven't sampled the blue enough yet to get a good estimate of its population. We need a better way to do that. So we're going to do it with ensembles of systems and coupling the runs. And if you look back to the previous figure, I had 4 colors picked in addition to the black. Turns out in the MD simulations, we sample 4 conformations. We do get the minor and the major, but we also get this weird intercalated and inverted structure. Now, if those existed experimentally at a population greater than 5 or 10%, you'd definitely see them. And these are not compatible with the NMR data that we have, so they're kind of anomalous structures or errors. And that's where we were until just recently. So if we get more sampling by using GPUs, lots of them on Blue Waters or other machines, essentially our prac is tying together these independent runs where we exchange information like temperature or changing force fields or pH, and that allows us to use lots of GPUs but also sample the space much more efficiently. And now with the replica-exchange, you can get this sampling of the blue just in 2 microseconds per. And this was originally with something like 30 replicas. Now it's up to, with multidimensional replica-exchange, it's up to 192 replicas. And we can get a distribution plot that shows kind of the percentage of conformations a particular distance from some A-form reference. So these 2 peaks here, these first ones are the experimentally populated structures. You can see they're much lower population than these anomalous structures. So there's clearly a problem with our force field. We've got to fix there. But we can show that 2 independent runs, very different initial conditions, give exactly the same results, which is nice. With multidimensional replica-exchange, we can do it even faster. And this is just showing overlap of principal component runs. So we can show that not only is the structural distribution right, but the modes of motion sampled are the same between independent runs. And what we do now in all our papers is we have scripts that enable you to do the analysis we do in our papers, so you can actually get our programs and figure out how to do it without reading the manual, if you want to do that. We also found out last year that we can go to a double time step in the MD integration just by shifting the mass of the hydrogens, or shifting the mass on the heavy atoms to the hydrogens. And we haven't shown any statistical difference we can get in the results we see as far as dynamics or the structural distributions in this, so we've routinely done this, and we hadn't anticipated we'd suddenly be able to double our simulation performance. It's kind of funny, you know, with a PRAC award, you're supposed to predict 3 years of your usage. So if I predicted it at my 2 femtosecond time step, suddenly I have twice as much computer time available because I'm going twice as fast. And so it's very often hard to predict exactly where these things are going to go, what problems we're going to encounter, what we're going to need to do to fix it, how long it's going to take. So now this is the same RNA tetranucleotide with the different colors are different force fields that are available out there, charm, different versions of amber. None of them well reproduce the peaks at the low are the ones that are representative of experiments. Some have some really weird ones like charm where some bases flip in a funny way. But we can now routinely generate these force field comparisons in about a day, what used to take us months to a year to do, thanks to access to the GPU resources. And what we are finding still is with these force field artifacts, sometimes when you think you're converged, you aren't necessarily converged. The longest replica-exchange simulations in 2013 on a UCG tetraloop were about half a microsecond each, and they found a nice population of the native structure. Well, if we crank those out in a much larger replica-exchange framework with better sampling, we now never populate the native state. We actually populate this anomalous conformation we call the ladder-like one, which is due to a problem with the chi parameterization. Now, other force fields that have purported to do very well for folding RNA structures is the Chen-Garcia that gets the experimental one right. It populates it more than the amber force fields do, but it does it via some tricks by altering the pairwise interactions between bases and bases without affecting the base-water interactions. It's called non-bond fix and charm. And we wanted to get away with trying to get these kind of good results without doing the non-bond fix. And so what I didn't mention is we can now fully converge the conformational distribution of RNA tetraloop, and we're doing that in various different cases. But what's most exciting is just results we've had in the past couple weeks that we knew from some papers by David Case that the phosphate radii were too small, so effectively the charge interaction was too tight. So if we increase the radii on that, it reduces the strength of that interaction. And also there's a new water model that got published. They call it the optimal point charge model. I think that's a little, I prefer off-point charge model. That's what it really represents. But Alexei Onofriev published that. Now, many postdocs have lost their careers to developing new water models that were never any better than the previous ones. So I was highly skeptical, and I asked my people to try it out. And suddenly what happened is it actually increased the populations of the expected experimental structures while decreasing the population of the non-experimentally observed structures. So if we consider kind of just the extreme of tip 3P with this greenish color going to the OPC in the black, you've reduced, with the regular amber force field, you've reduced the population of this inverted or intercalated structure, and you've increased the major and minor conformation. And now if you do that same thing with these new backbone modifications, you now get 3-to-1 population of the major to the minor, which exactly matches the NMR. And the population of this is low enough now that you wouldn't see it in NMR spectra. So we're kind of encouraged that the force field is actually giving decent results. And it's complex as to why it works, but it's a proper balance between the ions, the charge interactions, and others. We had known this previously because we had published results on generating ion models that were specific for each of these individual water models. And I don't know why I didn't think of it at the time, but what is RNA? It's just a polyanion. It's just a large string of negative charges. So it should be dependent on the water model. And it turns out results are a little better with TIP4P and even a lot better with this new off-point charge model. So we're fairly encouraged. We're now testing these. If Blue Waters can remain stable, we'll have all the tetraloop results done and other systems to verify this. I won't talk about this today, but we were very fortunate to receive one of these Ebola Rapid Awards, and this is going to be a super exciting project. Michael Kay's lab at the University of Utah in biochemistry has demonstrated this in HIV and also prototyped it in Ebola. It turns out there are these trimer proteins the virus produces that allow the virus to puncture cells and insert its genetic material. And Michael Kay has developed a phage technology that allows them to find peptides that bind to these membrane fusion proteins. And what's cool is it's a symmetric trimer. If you're lucky, you get 3 peptides to bind. They're all the same sequence. Then if you just link them together, you've got binding affinities that are tighter than can be measured due to the avidity on there. Now the problem they have is they select this with phage display. They're doing 12 mers. Phage display only has about a 1 in 2 million or 2 billion sequence coverage. So now we can go in a simulation and flip various places to see if these alter the binding affinity. We can also do it to map out the patent because we can see where things are variable and where things are not, and we need to have the particular structures. So we're just starting this up, developing the technology to run, extend these chains where they have structures for them. There's 2 structures already known with peptide bound for Ebola, so it's pretty exciting. Finally, I'm interested in the Blue Waters data sharing program. We're getting to the point where we want to make our results available to a large community. We've tried this before with the DNA results. I put it on my webpage. It got downloaded by spiders from China. That's about it so far. But we're going to try it with some of these ensembles that are converged so other people can test force fields and see if they can reproduce the same kind of results we do. And our analysis code's freely available and does much more than just molecular dynamics analysis, but it's described there, and I'll stop here for questions. Yeah?
How accurate and reproducible do you think our force fields are now for disordered polypeptides?
I know how they are for disordered nucleic acids, and they're very bad. So the amber force field way overstacks and way over intermolecular hydrogen bonds, whereas the charm is too extended. For disordered, none of us have really parameterized for that. So the current generation of force fields from charm and amber have started to look at side chain interactions and rotamer populations for bound states, but they haven't really done it in terms of the free state. So I would say that's a ripe area for research.
Any other questions?
Yeah.
Coming back to the 4 nucleotides RNA, so would you say that for DNA you would expect a better convergence or not?
That's a good question. So DNA, we get better convergence in duplex regions. The same is true in RNA in duplex regions. The problem with all of these is the interesting regions, which are loops, bulges, and single-stranded. So I would not say that DNA is better. And certainly at present, we have bifurcated in our force field optimization for DNA and RNA. They used to be very similar, and now they're breaking off. And I think they need to break off more. I think we need to separately parameterize purines versus pyrimidines and other stuff. So there's still some work to be done. This issue of balance with stacking versus intermolecular interactions and others is going to bite you in DNA, but not as bad because you don't have that O2' hydroxyl. But I wouldn't claim it's good yet.